Okay, let's unpack this. Imagine the very first cell. What was the absolute first thing it needed? To become distinct from everything else around it.
A boundary.
Exactly, a boundary. That boundary, of course, is the membrane. Today, we're diving deep into the intricate world of biological membranes and transport.
And we're drawing our insights straight from Chapter 11 of Lehninger Principles of Biochemistry, a classic text.
Right. Our mission here is to take what might seem like some pretty dense biochemical information, all those molecular mechanisms and pathways, and pull out the most important nuggets.
The core ideas.
Yeah, the core idea. So you can quickly grasp how these cellular gatekeepers are built, how they move dynamically, and how they control really precisely everything that enters and exits a cell.
Get ready for some surprising facts and maybe a few aha moments along the way. It connects to so much of cell function.
Absolutely. So let's start at the beginning. At its core, what is a biological membrane? Sounds simple maybe, but it's really not, is it?
No, it's beautifully complex. Fundamentally, it's a lipid bilayer. And this structure, this incredibly stable structure, it forms spontaneously in water, mostly from phospholipids and sterols.
Spontaneously, just like that.
Pretty much. What drives this amazing self-assembly, this tendency to just form a boundary, is something called the hydrophobic effect.
I remember that.
Yeah.
Water pushing oily things together?
Exactly. It's this powerful thermodynamic urge for nonpolar regions, like the lipid tails, to avoid water. It minimizes those unfavorable interactions. And that's why lipids with both polar and nonpolar parts, we call them amphipathic lipids, why they naturally form structures like micelles or bilayers or even hollow vesicles, liposomes.
Like tiny bubbles.
Yeah, and those vesicles likely resemble the very first primitive cells, creating that essential separation.
Okay, here's where it gets really interesting for me. It's not just a static barrier, right? It's always described as a fluid mosaic. What does that actually mean for how cells function day to day?
Right, the fluid mosaic model. It's absolutely key. It highlights that membranes are incredibly flexible, they can self-repair, and they're selectively permeable. They choose what gets through.
So not a solid wall.
Definitely not. Think of it more like a two-dimensional fluid, a sea. The lipids and many of the proteins within that bilayer, they aren't rigidly fixed in place. They can move laterally, sideways, within the plane of the membrane, almost like they're dancing around.
And that movement is important.
Oh, absolutely crucial. This fluidity allows for dynamic processes essential for a cell's life, like growth, movement, division, responding to signals. And it's not symmetrical either. Membranes have distinct sidedness. The inside face is different from the outside face.
How so?
Well, different types of lipids and proteins are precisely distributed across the two layers. For example, you typically find choline-containing lipids on the outer surface, while others, like, say, phosphatidylserine, are concentrated on the inner cytoplasmic side. That asymmetry is vital for signaling and recognition.
And cells aren't just one big bag either. They have all those internal compartments, organelles, like rooms within a house. How do membranes create those?
That's another major role. If we zoom out to the eukaryotic cell, you've got this extensive endomembrane system.
The Golgi, that network.
Exactly. The endoplasmic reticulum, Golgi apparatus, lysosomes, various vesicles, all defined and separated by their own specialized membranes. This whole intricate system synthesizes, modifies, and distributes membrane components and other cellular cargo throughout the cell. It's like a factory and postal service combined.
So things get shipped around.
Precisely. Proteins and lipids are trafficked from the through the Golgi. They get modified along the way, like glycosylation, adding sugar chains. And these modifications often act like molecular ZIP codes, dictating exactly where that protein or lipid needs to end up in the cell.
Okay, that brings us to the proteins themselves. They're not just floating icebergs, are they? What jobs do they do, and how do they actually integrate into this lipid landscape?
That's a critical point. The protein composition of a membrane directly reflects what that membrane does, its function.
So different membranes have different proteins?
Absolutely. Proteins are the real workhorses here.
Yeah.
They act as receptors, picking up signals from outside the cell. They're transporters, controlling what gets in and out. And they can be enzymes, organizing complex reaction sequences right there in the two-dimensional space of the membrane, which makes things way more efficient than if molecules were just floating randomly in the cytoplasm.
Okay, and how are they attached? You hear about integral and peripheral proteins.
All right, we classify them based on how tightly they're associated. Integral proteins are firmly embedded, often spanning the entire bilayer. You need pretty harsh convergence to get them out.
Stuck in there good.
Yeah. Then you have peripheral proteins, which are more loosely associated. They might bind to the membrane surface or to the integral proteins through weaker interactions like electrostatic forces or hydrogen bonds. And there's a fascinating third group, amphitropic proteins. These can actually switch between being membrane-bound and being soluble in the cytoplasm.
So they can move on and off the membrane.
Exactly. And that association is often regulated, maybe by phosphorylation or by attaching a lipid anchor. It allows the cell to control when and where these proteins are active at the membrane.
So how do scientists figure out, if a protein is integral, or how many times it snakes back and forth across the membrane?
That's a really important question in biochemistry. We can often predict the topology, how it sits in the membrane, just from its amino acid sequence.
From the blueprint.
Sort of. There's a common bioinformatics tool called a hydropathy plot. It scans the sequence looking for long stretches, maybe 20 to 25 amino acids, that are hydrophobic water-thering. A stretch like that is long enough and hydrophobic enough to likely span the lipid bilayer as an alpha helix.
Ah, so you count those hydrophobic stretches.
Basically, yes. A
Yeah.
protein might be monotopic, just dipping into one layer,
Mm-hmm.
or bitopic, spanning at once, like glycophorin in red blood cells, or polytopic, crossing multiple times, like bacteria hopsin, which famously crosses 7 times.
7 times.
And it's not always alpha helices. Especially in bacterially mitochondrial outer membranes, you find beta barrels, where the protein forms a barrel shape using beta strands. Porins are a good example. You also see interesting patterns like certain amino acids, TIR and TRAP, clustering right at the interface between the lipid tails and the watery environment, acting like anchors. And there's a positive inside rule. Positively charged residues like lysine and arginine tend to be on the cytoplasmic side.
Okay, we've talked about this fluid mosaic idea. Let's dive a bit deeper into the actual motion. What kinds of movement happen and what influences them?
Right, it's definitely dynamic.
Yeah.
The lipids themselves are constantly moving. They can exist in different physical states, sort of like the difference between liquid oil and solid fat. There's a more gel-like rigid state called the liquid-ordered low state, and a more fluid liquid-disordered LD state.
What determines which state it's in?
Several factors. Temperature's a big one. Higher temps mean more fluidity. Also, the type of fatty acid chains on the lipids. Longer, saturated chains pack tightly, favoring the ordered state. Unsaturated fatty acids, the ones with double bonds, have kinks in their tails. These kinks prevent tight packing, so they favor the fluid disordered state.
Makes sense. And cholesterol, you mentioned that before.
Cholesterol is fascinating. It sort of has a paradoxical effect. In membranes with lots of unsaturated fats, which are already fluid, cholesterol tends to restrict motion a bit, making them slightly more ordered. But in membranes with mostly saturated fats, which tend to be more gel-like, cholesterol actually disrupts the tight packing, increasing fluidity. It acts like a fluidity buffer, keeping the membrane in a good functional range across different conditions.
Okay, so the lipids themselves are moving. What about movement within the bilayer?
Yeah, 2 main types. Lateral diffusion, moving sideways within one layer or one leaflet of the bilayer, is incredibly fast.
How fast?
We can measure it with techniques like FR rate fluorescence recovery after photobleaching. A lipid molecule can basically zip across the surface of a typical eukaryotic cell in just a few seconds. It's really rapid.
Wow, but what about moving from one side of the membrane to the other?
That's transbilayer movement, or flip-flop. That is very slow if unaided. Think about it. The polar head group of the lipid has to be dragged through the hydrophobic, oily core of the membrane.
Doesn't want to do that.
Exactly. It's energetically very unfavorable. The activation energy barrier is huge, so spontaneous flip-flopping is rare.
But it must happen sometimes, right? Like when new lipids are made, they need to get to the right side.
Absolutely. And cells don't leave it to chance. They have specialized protein machines, enzymes, called phospholipid translocators to manage this.
Okay, what do they do?
There are different types. Flipases typically move specific lipids from the outer extracellular leaflet to the inner cytoplasmic leaflet, and they usually consume ATP for energy. Flopas generally move them in the opposite direction, also often ATP dependent. Some belong to that big ABC transporter family we'll talk about later. And then there are scrambloses. These don't usually need ATP. They move lipids down their concentration gradient, helping to randomize or scramble the distribution, often activated by signals like an increase in calcium ions.
So the cell actively maintains that asymmetry we talked about.
Precisely. This controlled enzyme-catalyzed movement is crucial for establishing and maintaining the specific lipid composition of each leaflet, which, as we said, is vital for function. Messing it up can even be a signal for programmed cell death apoptosis if phosphatidylserine gets exposed on the outside, for instance.
Right. Now, what about these membrane rafts? You hear that term a lot. How do they fit into this dynamic picture? Are they like little islands?
That's a good analogy. Even within a single leaflet, the lipids aren't always uniformly mixed. Membrane rafts are thought to be transient, specialized microdomains, little fluctuating patches.
What makes them special?
They tend to be enriched in certain lipids, particularly sphingolipids and cholesterol. These lipids often have long, straight, saturated fatty acyl chains that can pack together very tightly with cholesterol. This makes these raft regions slightly thicker and less fluid, more in that liquid-ordered state compared to the surrounding membrane.
And does that matter functionally?
Well, definitely. This difference in thickness and fluidity leads to a kind of segregation. Proteins with shorter transmembrane segments might be excluded from rafts, while others, maybe those with longer segments or specific lipid anchors like GPI anchors, tend to cluster in the rafts.
So they bring specific proteins together.
Exactly. Rafts are thought to be crucial for organizing signaling complexes. By bringing specific receptor proteins and downstream signaling molecules together in close proximity, they can make signaling much more efficient. They can make up a significant portion of the cell surface too. And they're not static islands, they're dynamic. Proteins can move in and out, but for short periods, they function as organizing platforms.
Is there a specific type of raft?
Yes, one well-studied type is the catiolae. These are small, flask-shaped inward dimples or invaginations of the plasma membrane. They're associated with specific integral proteins called caveolins, which actually seem to help force the membrane to curve inwards like that. Caveolae are involved in membrane trafficking, endocytosis, and cellular signaling.
This brings up curvature and fusion. It sounds like shaping the membrane and getting membranes to merge must be really important.
Incredibly important. Membrane curvature and membrane fusion are fundamental to countless biological processes. Think about vesicle budding during transport, endocytosis, exocytosis, like releasing neurotransmitters, even cell division and viral entry.
How do cells control the shape?
There are proteins specialized for this. Some proteins, like those with bar domains, can sense or even induce membrane curvature. They might insert a curved part of the protein into one leaflet or wedge an amphipathic helix into the lipid packing, forcing it to bend.
And fusion, like when a vesicle releases its contents.
Fusion is a highly orchestrated, energy-requiring process. It doesn't just happen spontaneously. It's mediated by specific fusion proteins. The best studied examples are the snares. Yes, you typically have V snares on the vesicle membrane and T snares on the target membrane, like the plasma membrane. These proteins have helical domains that recognize each other and coil together, forming a tight bundle. This coiling process literally pulls the two membranes incredibly close together, overcoming the repulsion between them, disrupting the bilayers locally, and initiating the fusion event.
Wow, they physically pull them together.
They do. It's a remarkable mechanical process at the molecular level. And this mechanism is so crucial, especially in neurons, that it's the target of some really potent neurotoxins.
Like Botox.
Exactly. Botulinum toxin and also tetanus toxin work by cleaving specific snare proteins. This prevents neurotransmitter release, leading to flaccid paralysis, Botox, or rigid paralysis, tetanus. It highlights how vital snare-mediated fusion is.
Okay, stepping back a bit. Beyond these internal dynamics, how do cells use their membranes to interact with the outside world and with each other? Building tissues, for instance.
That's another critical membrane function. Cell-to-cell adhesion and interaction with the extracellular matrix, the scaffold outside cells, are mediated by integral membrane proteins.
Like what?
Well, you have integrins. These are really important transmembrane receptors that link the cell's internal cytoskeleton to the extracellular matrix. They also act as signaling molecules, transmitting information in both directions across the membrane. Then there are cadherins. These primarily mediate cell-to-cell adhesion, often through homophilic interactions, meaning a cadherin on one cell binds to an identical cadherin on the neighboring cell. This is crucial for holding tissues together. And selectins are involved in transient cell adhesions. They bind to specific carbohydrates on the surface of other cells, playing key roles in things like immune cell trafficking and blood clotting.
All right, so we've built the house, seen how its walls are fluid and dynamic, how rooms are made, and how it connects to the neighbors. Now let's talk about the doors and windows. Transport. How does the cell decide who gets in and who stays out? This seems absolutely critical.
It is absolutely critical for life. Every single cell needs to bring in nutrients, get rid of waste products, and maintain very specific concentrations of ions inside, homeostasis. Now, a few small nonpolar molecules, think oxygen, CO2, maybe some steroid hormones, they can just slip through the lipid bilayer directly by simple diffusion, down their concentration gradient.
But most things can't do that.
No, definitely not.
Yeah.
For almost any polar compound, sugars, amino acids, nucleotides, or any ion like sodium, potassium, calcium, the lipid bilayer is basically impermeable. They need help.
So proteins again?
Proteins again. Specific membrane protein carriers or transporters are essential, and the scale is just huge. The human genome, for instance, encodes something like 2,000 different transport proteins.
2,000. Just for moving things across membranes.
Yep. It really underscores how vital and complex this process is. It touches on everything from nutrient uptake to nerve signaling to energy metabolism.
Okay, let's get the basic category straight. What's the fundamental difference between passive and active transport? You mentioned gradients.
Right. The key distinction boils down to energy and the direction of movement relative to the electrochemical gradient. Passive transport, which is also called facilitated diffusion, simply helps the solute move down its electrochemical gradient.
So from high concentration to low.
Exactly. Or for ions, it also considers the electrical potential across the membrane. It's like rolling downhill. The protein just provides an easier path, increasing the rate of movement, but it doesn't require the cell to expend metabolic energy directly because the gradient itself provides the driving force.
Okay, and active transport.
Active transport is the opposite. It moves solutes against their electrochemical gradient, pushing them uphill, essentially, accumulating them on one side. This is thermodynamically unfavorable, like pushing a rock uphill, so it requires an input of energy.
Where does that energy come from?
It can come directly from a chemical reaction, most commonly the hydrolysis of ATP. That's called primary active transport. Or the energy can come indirectly. The cell might use energy to pump one solute uphill, creating a gradient, and then the downhill flow of that solute is coupled to the uphill movement of another solute. That's secondary active transport. It uses energy stored in an existing gradient.
Okay, that makes sense. Energy is the key. Now, within these categories, you mentioned transporters and channels. How do they differ in their mechanism? How do they actually get things across?
Good question. Both transporters and ion channels work by lowering the activation energy for moving a solute across the membrane, making it much faster than unassisted diffusion. But their mechanisms are quite different. Transporters, sometimes called carriers or primases, function more like a revolving door or an airlock. They bind their specific substrate, their cargo, with high specificity, almost like an enzyme binding its substrate. Then they undergo a conformational change, a shape change, that exposes the binding site to the other side of the membrane, releasing the substrate. Critically, a transporter typically has two gates, and they are never both open at the same time.
One side or the other, but not straight through.
Exactly. This prevents leakage.
Yeah.
And because they have this binding and conformational change cycle, transporters are saturable. Just like enzymes, there's a maximum rate, Vmax, at which they can work, because there are a finite number of transporter molecules, and each takes time to complete its cycle.
Can you give an example?
Sure. A classic example is GLUT1, the glucose transporter found in red blood cells and many other tissues. It facilitates glucose entry down its concentration gradient, but does so about 50,000 times faster than glucose could diffuse across the lipid bilayer on its own. It's highly specific for D-glucose, and it shows saturation kinetics. At typical blood glucose levels, GLUT1 is working close to its maximum rate. There are many GLUT transporters like GLUT4 in muscle and fat, whose activity is boosted by insulin, allowing those cells to rapidly take up glucose after a meal.
Okay, so transporters are like specific revolving doors. What about ion channels?
Ion channels are fundamentally different. They form more of a simple pore or tunnel through the membrane. When the channel's gate is open, ions can flow through extremely rapidly, almost as fast as they can diffuse through water.
Much faster than transporters.
Orders of magnitude faster. We're talking millions, even hundreds of millions, of ions per second passing through a single open channel. They generally show some specificity for particular ions, like being a potassium channel or a sodium channel, but they are not saturable in the same way transporters are. The flow rate mainly depends on the ion concentration gradient.
What controls the gate?
The gates are regulated. They aren't just always open. They open and close in response to specific biological signals. Some are ligand-gated channels, meaning they open when a specific molecule, a ligand like a neurotransmitter, binds to them. Others are voltage-gated channels, which respond to changes in the electrical potential difference across the membrane. These are absolutely essential for generating nerve impulses.
Right, the action potential relies on those voltage-gated channels opening and closing in sequence.
Precisely. And scientists can actually study the tiny electrical currents flowing through single-ion channels using a technique called patch clamping. It lets us observe the behavior of individual channel molecules opening and closing.
Fascinating. Let's circle back to active transport, the uphill battle. What are the main types of pumps that use energy, often ATP, directly?
Okay, the primary active transporters. There are several major families. One very important group is the P-type ATPases.
P-type.
Yes, because they get reversibly phosphorylated. An inorganic phosphate group from ATP gets attached to them as part of their pumping cycle. They primarily transport K applications, positive ions. The absolute classic example found in virtually all animal cells is the DnaA plus K plus ATPase, often just called the sodium-potassium pump.
Ah, the one that uses tons of energy.
That's the one. For every molecule of ATP it hydrolyzes, it pumps 3 sodium ions out of the cell and 2 potassium ions in, both against their respective concentration gradients.
3 out, 2 in. That changes the charge balance too, right?
It does. Because it moves unequal amounts of charge, it's electrogenic, meaning it contributes directly to the electrical potential across the plasma membrane. This pump is responsible for maintaining those steep DnaA plus and K plus gradients that are essential for nerve impulses, muscle contraction, maintaining cell volume, and driving secondary active transport. And yes, it's incredibly energy hungry, consumes maybe 25% or more of a human's total energy expenditure at rest.
Wow. Any other P-type examples?
Sure. The Circa pump in muscle cells is another P-type ATPase. It pumps calcium ions from the cytoplasm into the sarcoplasmic reticulum, which is essential for muscle relaxation after contraction. There are also H+ pumps in plants and fungi, and H+ K+ pumps in our stomach lining that acidify stomach contents.
Okay, what about other ATPase families?
Then you have the V-type and F-type ATPases. They look structurally different from P-types and work a bit differently, primarily pumping protons, H+. V-type ATPases are found in vesicles and vacuoles, hence the V, and membranes of organelles like lysosomes. They pump protons into these compartments, acidifying them, which is important for lysosomal enzyme function, for example. F-type ATPases are really interesting. They're found in bacteria, mitochondria, and chloroplasts. They can pump protons using ATP, but crucially, they often work in reverse.
In reverse? How?
They act as ATP syntheses. They harness the energy stored in a proton gradient created during cellular respiration or photosynthesis, allowing protons to flow back down their gradient through the F-type ATPase and using that energy to synthesize ATP from ADP and phosphate. They are the primary ATP producers in most cells. So they have this F designation, often linked to factor.
So they can build ATP using a proton gradient. Amazing. Any other major pump families?
Yes, one more huge and incredibly diverse family. The ABC transporters.
It stands for ATP binding cassette. These transporters all share a common core structure that includes domains which bind and hydrolyze ATP to power transport. They transport an absolutely vast array of substrates, ions, amino acids, peptides, lipids, bile salts, toxins, drugs, you name it. Mostly they pump things out of the cell or into organelles against concentration gradients.
How many are there?
There are at least 48 known ABC transporter genes just in humans. They play roles in nutrient uptake, toxin removal, cholesterol transport, and unfortunately, in drug resistance.
Drug resistance?
Yeah. One famous example is the multi-drug transporter, or MDR1, also called P-glycoprotein. Cancer cells can sometimes overproduce this protein, which actively pumps a wide variety of chemotherapy drugs right back out of the cell, making the tumor resistant to treatment.
That's a major challenge.
It is. There are other ABC transporters involved in drug resistance, too. Interestingly, while most ABC transporters are pumps, there's one famous exception, the CFTR protein.
The one involved in cystic fibrosis. That's right. CFTR is structurally an ABC transporter. It binds ATP, but it actually functions as a regulated chloride ion channel. Defects in CFTR cause the thick mucus characteristic of cystic fibrosis because chloride and water transport are disrupted.
Okay, so primary active transport uses ATP to create gradients, like the DnaA plus gradient. You said cells can then use that stored energy, secondary active transport.
Exactly. It's like charging a battery with primary active transport and then using that battery power for other tasks via secondary active transport. The steep electrochemical gradient for sodium ions across the plasma membrane, high outside, low inside, maintained tirelessly by the DnaA plus K plus pump, is a major energy source for animal cells.
How does it work?
A secondary active transporter binds both the ion that's flowing downhill, like Na+, and the solute that needs to be moved uphill, like glucose. The favorable movement of Na+ down its gradient provides the energy to drive the unfavorable movement of glucose against its gradient into the cell.
So they move together.
Often, yes. If both solutes move in the same direction, it's called symport, like the No plus glucose symporter in intestinal and kidney cells. If they move in opposite directions, one in, one out, it's called antiport, like the chloride bicarbonate exchanger in red blood cells, which is crucial for CO2 transport.
And this can really concentrate things inside.
Oh, absolutely. The No plus glucose symporter, for example, can use the potent No plus gradient to accumulate glucose inside intestinal cells to concentrations thousands of times higher than in the gut lumen. This ensures we absorb virtually all the glucose from our food.
And you mentioned kidney cells, too.
Yes, a similar No plus glucose symporter is responsible for reabsorbing glucose from the filtrate back into the blood in the kidneys. And interestingly, a new class of drugs for type 2 diabetes, the gliflozins, work by inhibiting this kidney transporter.
How does that help?
By blocking glucose reabsorption, they cause excess glucose to be excreted in the urine, which helps lower blood glucose levels. It's a direct application of understanding secondary active transport mechanisms.
Clever. Okay, one more simple molecule. Water. It seems like it should just diffuse, but does it need help too?
It does, especially for rapid movement. While some water can diffuse directly across the lipid bilayer, it's relatively slow. For rapid bulk flow of water, cells use specialized channels called aquaporins, or AQPs.
Water channels.
Exactly. These are integral membrane proteins that form pores specifically permeable to water molecules. They're found in all kingdoms of life.
Why are they needed?
They're crucial in tissues where rapid water movement is essential. Think kidney tubules, where vast amounts of water need to be reabsorbed. Or red blood cells, which need to swell or shrink rapidly in response to osmotic changes without bursting. Aquaporins allow water to move incredibly fast, maybe 1 billion water molecules per second through a single channel.
1 billion per second, that's staggering.
It is. And a key feature is that they are highly specific for water. They have structural features that prevent the passage of ions, especially protons, H+. Letting protons through would dissipate the vital electrochemical gradients across membranes. So aquaporins have evolved to be strictly water selective.
Okay, to wrap up transport, let's revisit ion channels and their amazing specificity. You said the potassium channel lets potassium through way better than smaller sodium ions. How on earth does it manage that? It seems backward.
It really is a marvel of molecular engineering, explained beautifully by structural biology. Let's take that bacterial K+ channel as the model. It has a narrow part called the selectivity filter.
The gatekeeper part.
Precisely. This filter is lined with specific atoms, carbonyl oxygen atoms, from the protein's own backbone, arranged in a very precise geometry. Now, ions in water are normally surrounded by a shell of water molecules. They're hydrated.
Yeah.
To pass through the narrow filter, an ion has to shed most of these water molecules. This costs energy.
Right. Takes energy to pull the water off.
Yes. But for a potassium ion, K+, as it enters the selectivity filter, those precisely positioned carbonyl oxygens perfectly mimic the hydrating water molecules it just lost. They form ideal coordination bonds with the K+ ion, essentially replacing the water shell and stabilizing the ion as it passes through. The energy cost of dehydration is perfectly compensated by the favorable interactions within the filter.
Okay, so it fits perfectly. What about sodium? It's smaller.
And that's the key. A sodium ion, now plus, is indeed smaller. When it tries to enter the selectivity filter, it's too small to make simultaneous optimal contact with all those carbonyl oxygens. The filter can't collapse around it effectively, so the energy compensation isn't good enough. It costs too much energy for NAB+ to dehydrate and enter this filter that's perfectly sized for K+ ion. As a result, K+ passes through about 10,000 times more readily than NAB+I, despite NAB+ being smaller.
Wow. So it's not just size exclusion. It's about perfect energetic compensation for dehydration.
Exactly. It's an incredibly elegant mechanism that allows for both high specificity and very rapid throughput for the selected ion. A beautiful example of structure determining function at the molecular level.
So reflecting on all this, what's the big picture? We've gone from the basic lipid bilayer architecture, driven by simple hydrophobic forces, through its fluid dynamics and asymmetry, all the way to these incredibly sophisticated protein machines, transporters, pumps, channels that act as the cell's gatekeepers.
It really showcases how fundamental membrane biology is. These molecular mechanisms, the thermodynamics of lipid assembly, the kinetics of transport, the integration of protein function within the lipid environment, they underpin almost everything a cell does.
Yeah, think about how this intricate dance of lipids and proteins at the membrane level enables everything from nerve impulses firing in your brain, to your muscles contracting, to nutrient absorption, energy production via ATP synthase, and even how your immune system recognizes friend from foe.
It's all happening constantly, billions of times per second, in every one of your cells. These molecular gatekeepers, operating with incredible precision and efficiency.
It makes you wonder, what other everyday biological phenomena are secretly governed by these membrane dynamics and transport processes that we haven't even fully uncovered yet? We hope this deep dive has given you, our listener, a new appreciation for this complex and absolutely vital world happening within and around every single cell.
It's a foundation of biochemistry and cell biology.
Absolutely. Thanks for diving deep with us today and for being part of the Last Minute Lecture family.